Hi, everyone. We are going to talk about the start of our new unit, Unit 5, Heredity. And to do that, we're going to be talking about these big ideas about how genes can get passed on from parents to offspring. And in order to do that, we have to talk about chromosomes. And in order to do that, we need to talk about meiosis. And so meiosis is this idea, is the formation, the big idea here. There we go. Formation of gamete cells in sexually reproducing organisms. So this is all about how complex organisms, you want to think about like animals, plants, fungi, eukaryotic organisms, most of them are going, all of them really are going to be in some way sexually reproducing. And so how do they make those sex cells or gametes that will produce offspring? And so we'll be taking a look at gametes. If you wanted a two-word definition for meiosis, all it is making gametes. And so we'll talk about the differences here, but gametes are haploid cells that come from diploid cells. So let's take a look at that. So all cells can be kind of categorized as diploid or haploid. And for the most part, every cell we've talked about has been diploid. And what that means is that there are going to be two full sets or pairs of chromosomes called homologous pairs, or homologous chromosomes. Now, these are going to, the homologous chromosomes are going to have the same shape and size and will have the same genes, but they won't necessarily have the same expression of those genes or have the same allele, for example. We get homologous chromosomes, one from each parent, right? 50% of our DNA from mom and dad, and we use the symbol 2N to represent a diploid cell. Body cells, also known as like somatic cells, are diploid. Skin cells, leaf cells, any cells you take from most of our body is going to be a diploid and or somatic cell. Haploid cells are cells that contain only one set of chromosome, meaning every chromosome in that nucleus is its own unique chromosome. There's no other chromosome that has the same genes on it. There's no other chromosomes with the same shape or size. And so for haploid cells, we use the symbol N to represent that. So really, the difference is just between whether or not there are homologous chromosomes. I would say that's like that is the key definition of whether it's a diploid or haploid cell. Okay? And so gametes, these sex cells that are going to be produced from meiosis, are going to be haploid cells. And so we're going to see egg cells in females and then sperm cells or pollen in males. And so haploids, again, one copy of each chromosome. As soon as we have these twins coming about, right, these are going to be our homologous pairs. And so when we say they have the same gene, they might have a gene for hair color here, right? And maybe this chromosome, the middle one, maybe this one has something for eye color here. But they don't have to have the same alleles. So we'll talk about dominant and recessive in a future topic, but maybe this one has a dominant allele for hair color for brown. Maybe this one has a recessive allele for blonde. And maybe, this would express a brown phenotype from those two homologous pairs. And then this one, they would have the same alleles, but they just inherit those from mom and dad. And so this is a way, when we talk about genetics and Punnett squares, we want to think about this, right? The genes that we always talk about, genes are our DNA, and they determine the proteins that get built. These are found on the chromosomes. And we need to find where genes are, because we know where all of the genes are and what chromosomes they're found on, right? Humans have 46 total chromosomes, 23 pairs, and so we use chromosomes pairs 1 through 23, and we can find what genes are found on what chromosomes. And so kind of the flip side of meiosis is the production or making of offspring, and so that's taking those two gametes that are produced from meiosis and fertilizing them. So this is just called fertilization. All right, two haploid gamete cells come together in sexual reproduction to produce a diploid cell. So we have a haploid sperm cell here, male. There are 23 chromosomes in here. Haploid egg cell, 23 chromosomes in here. Fertilization takes place, and now we have a zygote 46 chromosomes, which is standard, that will undergo their stem cells that'll form, and mitosis will keep happening to develop into a offspring, a full adult. Okay. And so just coming back to meiosis, meiosis results in 4 daughter cells. These are all important things we have to remember for this unit. With half the number of chromosomes as the parent cell, here we're going from diploid to haploid. Okay, so we say produces 4 haploid daughter cells or sex cells called gametes. And just maybe a clarification, these diploid parent cells, sometimes you'll see them, these are called like germ cells. These are diploid cells that have the ability to undergo meiosis and become or create these gametes. There are 2 cell divisions in meiosis. These are meiosis 1 and meiosis 2. And so we're going to take some time to talk about each of those. And so meiosis 1, you want to just think about like our cell division. So we have 1 cell division to have 2 cells. We have, this is the second cell division. So this is cell division 1. This is cell division 2. And so 2 cuts should equal 4 cells. Right? This is just meiosis. And so meiosis 1, that first step, this is the part that is going to, we want to be careful about what is separating in each meiosis. So for example, in meiosis 1, this separates homologous chromosomes. Homologous chromosomes, this is like our big idea for the unit. And this is separating our homologous chromosomes. And so what we'll do is we'll do 1 homologous pair. And all we're doing in meiosis 1, not all we're doing, but we're going to be taking our homologous pairs and moving them to different chromosomes. And so an important thing to know about this is how we go from 2N to N, right? Because we've broken up the homologous chromosomes, we've broken up the twins. And so this goes from a diploid cell to haploid cell. So this happens, this reduction from diploid to haploid takes place in meiosis 1. And then a lot of stuff we're going to see here sounds very similar to mitosis because it's very similar, it's the same steps, with some key differences we'll talk about later. But in prophase 1, we have all of these things that happen in mitosis. Nuclear envelope begins to disappear, spindle fibers begin to form, our DNA that has been duplicated already begins to condense into chromosomes made up of sister chromatids. Double chromosomes pair up based on size, shape, centromere location, genetic information, so that we can separate those homologous pairs. And then we have this other term, this new term, this is called synapsis. This is the connecting of homologous chromosomes to form a tetrad. And so here we have a homologous chromosome. Synapsis would be connecting in the middle here. And then it's called a tetrad just because tetra means 4. We have 1, 2, 3, 4. We have 4 sister chromatids, which will eventually be their own chromosomes in different cells. Okay, so each homologous pair, eventually these will be 4 cells, and each sister chromatid will go into each of those cells. And so what happens, this takes place in prophase 1, this is called crossing over. So this is where homologous chromosomes are lined up together, they've just condensed, and they're exchanging genetic information. And this is going to be a way to increase genetic diversity in organisms. Okay, so this is going to be a big idea. Another highlight of this unit is to talk about ways that organisms or sexually reproducing organisms can increase genetic diversity in populations. Crossing over is one of them. And so we can see that taking place over here. We have our chromosomes, our homologous chromosomes, they overlap, and now they have exchanged parts of their chromosomes with each other. So again, they still have the same genes, right? So at the same spot of their chromosomes, this is called the location of the genes. This is called the loci or loci. So maybe this blue homologous chromosome has big A, and since it's a replicated half, this would also be big A. Maybe this red one has the recessive allele for little a. Crossing over takes place, and now this homologous pair now is big A, little a, big A, little a. And that's going to become a factor once the cell continues to divide and form these 4 gametes. Metaphase 1 takes place, so our chromosomes are just lining up on the metaphase plate or equatorial plane, as we sometimes will see it. Our spindle fibers need to attach to the middle parts of these homologous chromosomes in order to separate them. And then here we can kind of see this is a model of a chromosome. These are just like examples of where different genes can be found on the chromosome. And so if crossing over is more likely to happen at the extreme ends of a chromosome than it is in the middle. So for example, we could say genes A and D, these 2 extreme ones, are more likely to cross over than say like C or B or D. So anaphase 1 is happening, and so again, anything in meiosis 1, this is all about homologous chromosomes. Spindle fibers separate chromosome pairs. So if you see the word homologous chromosomes and talking about meiosis, it has to be about meiosis 1. Emphasis on that. Each double chromosome migrates to opposite sides of the cell. Telophase 1, nuclear envelope reappears at 2 sides. Each nucleus contains 1 double chromosome from each pair. Nucleus only contains half the total from the parent nucleus. And so we'll see this happen. Cytokinesis will split or separate the cell into 2 daughter cells and begin to uncoil. That's chromatin, right? That's just our spaghetti bowl of DNA and histone proteins, just all uncondensed, floating inside our nuclei. And so the end result of meiosis 1, the daughter cells are haploid and genetically unique from each other. Okay, that'll be important. And then meiosis 1 is known as the reductive phase because we are going from diploid to haploid. And so this is the part, this is where we want to remember that diploid to happens in meiosis 1, which is going to be this part of the diagram. And so you can see prophase 1, homologous chromosomes are separating to opposite sides, and now 2 cells have formed. So let's take a look at meiosis 2. And so in meiosis 2, we have separated our homologous chromosomes, so we cannot do that again. This time we're separating our sister chromatids. All right, we're taking our X and we're splitting them up to opposite sides. So prophase 2, metaphase 2, anaphase 2, I'm going to kind of skip through these because, again, they're all the same things that are happening. Meiosis 2, important to note, this is similar to mitosis because we saw mitosis is just a splitting or separating of sister chromatids. Mitosis does the same thing. I guess we could say a difference, though, between mitosis and meiosis 2 is meiosis 2 is going from end to end. So in this case, we could say it's going from 23 chromosomes to 23 chromosomes. Mitosis goes from 2N to 2N, right? We're staying diploid here. And so for humans, that's going to be 46 chromosomes to 46 chromosomes. Okay, so they're maintaining their ploidy number, how many types of chromosomes they have in them. And they're doing that by separating the separating the sister chromatids, not the homologous chromosomes. So anaphase 2 separates those. Telophase 2, nuclear envelopes reform. Chromosomes begin to uncoil back into chromatin. And so cytokinesis is going to split these cells up. The end result are daughter cells that are haploid and genetically unique from each other. So we can kind of see a very simplistic drawing of meiosis happening here. Interphase, we have our duplicated chromosomes. Prophase 1, we can see homologous chromosomes pair up, forming a synapse. Crossing over will take place. Once that happens, these homologous chromosomes will go to the metaphase plate. Spindle fibers are attached to the kinetic cores or the centromeres of the chromosomes. Anaphase 1 are homologous chromosomes. Let's clarify that. Homologous chromosomes are pulled apart 2 new nuclei are formed. Metaphase 2, anaphase 2. This time we're doing sister chromatids. All right, so now we're breaking up those X's. And then our end result are 4 gametes, right? 4 daughter cells. These are going to be sex cells, sperm cells in males, egg cells in females. All right, so one other thing to kind of note is that meiosis in males and females are going to look slightly different. So males, they go through this process called spermatogenesis. So it's really just in 2 words, meiosis in males. And so for sperm cells, it's very standard. It actually kind of just looks like what meiosis is over here. We just have 2 cell divisions. We should get 4 daughter cells, each with, if we were saying this was like a dog, which has 78 chromosomes, here we would have 39, and then each gamete should have 39 in each sperm cell after that, right? Because we go from 2N to N to N. If we look at this in meiosis in females, this is a slightly different process. And so if we maybe create some space here, female meiosis is called oogenesis. Okay, and so what they do is they have a cell, a germ cell that will undergo meiosis. And what happens is they will basically invest all of their resources and nutrients in one cell. And so they'll have a big cell here, and then they'll have like a very tiny cell here. This is called a polar body. The reason the polar body exists, and it'll eventually degrade and not do anything, is to maintain our chromosomal integrity or maintain our chromosomal number. If this is a lettuce cell, there are 18 chromosomes in a lettuce cell. Here we're still going from 2N to N. So we need 9 chromosomes in here and 9 chromosomes in here. That polar body breaks up, and so they will not be a part of the second division, meiosis 2. This time it makes another polar body and one final cell. Right, and so we still have N here chromosomes. We should have 9 and 9 in each one. Right, and so this is an egg cell. The egg cell, compared to the sperm cell, the egg cell has all of the organelles. It has all of the nutrients. It's the majority that will make up the zygote once fertilization takes place. And so these polar bodies exist just as a way to basically divert the chromosomes away from the egg cell. So meiosis in females really just will technically make like 3 daughter cells, but really we're just interested in one, and that's the egg cell or oocyte. Okay, so you'll see those terms come up, oogenesis and spermatogenesis, and kind of give some context to that. All right, so let's take a look at some differences between mitosis and meiosis. So both have DNA coil into chromosomes. Both have nuclear envelopes disappear in prophase. Both use fibers to separate chromosomes, spindle fibers. If that doesn't happen, that will lead to what we call non-disjunction. Both have nuclear envelopes reappearing, right? That's in telophase. I guess we should say that's anaphase as well. Both have chromosomes uncoil, and that'll lead into like interphase or telekinesis and cytokinesis. And then both are followed by cytokinesis and the production of daughter cells. So I would just start this. You want to be familiar with our 2 types of cell division, mitosis and meiosis. All right, so those are some similarities. Let's talk about some differences. So mitosis makes, 2 daughter cells. Versus meiosis, which will make 4. Mitosis is going to go from diploid to diploid, which we could say 2N to 2N. Maybe we could also say 46 to 46 in humans. This is different from meiosis, which goes from diploid to haploid, 2N to N. And if this was humans, 46 to 23 chromosomes. For mitosis, we have one cell division, one cut. Meiosis is 2 cell divisions. Mitosis, this is for growth and development. In somatic cells. And for meiosis, this is for gamete production. And meiosis only occurs in sex organs. So that's testes for males and the ovaries for females. And then lastly, mitosis is going to be genetically identical offspring, right? We're basically making clones, genetically identical. We want them to be identical, right? Body cells don't want to change. That would cause errors in DNA replication or errors in cell division, which could lead to tumors or cancer forming. Meiosis, so we want to create genetically unique offspring. Otherwise, us and all of our siblings would be the same. And so these are genetically unique cells. Okay, so I would encourage you take a look at